Now we are going to enter a new chapter of botany area. The chapter is anatomy of flowering plants. Actually, anatomy is the study of internal structure. As we studied morphology, morphology was the study of outside or peripheral structures. So, but anatomy means internal structure. Study of the internal structure is known as anatomy. Here we will study the structure of the organ. The internal parts are made up by organs. Different organs are there in the inside part of the body, and these organs are made up by different types of tissues, and each and every tissue is made up by different type of cells. So the study of cell is known as cytology, study of tissue is known as histology, and the study of morphological feature is known as or external feature is known as morphology, and the study of internal structure is known as anatomy. So now we will study the internal structure of plants, including monocot plants and dicot plants. But in both cases the anatomy is different because of the different arrangement of tissues in both of the cases. So now we will study the different tissues with related to the development and formation of different organs in the body of plants. So firstly we are going to see the tissue. Actually, the term tissue was coined by one scientist whose name is Bichat. So who introduced the term tissue? Tissue is a term used to describe a group of cells which are having common origin and common function, and sometimes common structure also. So a tissue is a group of cells with a common origin and a common function. That is a general simple definition of a tissue. Tissue is a group of cells having common origin, that means developmental stages. Its common origin and having common function. So such a type of group of cells is known as tissue. So tissue, different types of tissue we can see in plant body. So generally we can classify the tissue into two categories. Actually, meristematic tissue and permanent tissue. Actually, meristematic tissue will be giving rise to the permanent tissue. That means permanent tissues are formed from meristematic tissue. So meristematic tissue is the producer of all types of cells. Now we will study its classification. Again, this meristematic tissue is divided into different subcategories, such as primary meristem and secondary meristem. Primary meristem and secondary meristem. Like that, permanent tissue is also divided into two categories: simple tissue and complex tissue. Simple and complex. Now we are going to enter the details of meristematic tissue. So meristem is actually a term used to describe a group of tissue or cells which are having the high rate of division capacity. That means they are the fastly dividing cells. So characters of the meristem we will study. So meristem is fastly dividing cells. Fast dividing. So meristems are fastly dividing cells. They are active cells, but they are having high rate of mitosis property, that means division process. So they are having actively involved or cytoplasm as well as many cell organelles. So but they are immature cells and they are having less property of differentiation. That means they are young cells. They are fastly dividing young cells. So actually this meristem is the firstly formed cells in the seed. We know that a new plant is generated from the seed. In the seed, embryo axis is present, cotyledon is present, and seed coat is also present. But main part is embryo axis. Embryo axis or embryo is made up by special type of meristem cells. That meristem is known as actually promeristem. So what is pro-meristem? Pro-meristem means first meristem. First meristem. Where first meristem is present? First meristem means the meristem cells or tissue which are present in the embryo. So embryo will be at the time of germination, this embryo cells or this pro-meristem will be divided to form the tissues, other tissues that are responsible for the growth and elongation of the plant body. So pro-meristem means the meristem which is present in the embryo. Then after that this meristem is divided to produce different plant body, different plant parts such as shoot part and root part. So other characteristics of the meristem include they are having actually dense cytoplasm. Dense cytoplasm, then small vacuole, and then they are having one active nucleus. Active nucleus is also there. Then its cell wall is very thin. That is another character. Thin cell wall. Okay. Thin cell wall is present. Then they are actually meristem cells are present in the apices of shoot and root. That means at the apex of shoot part and apex of root part. So such meristems are known as apical meristem. So again another category. Second type of meristem is known as apical meristem. So now we are going to see the classification of the meristem. So meristem is divided into two categories: primary meristem and secondary meristem. Primary meristem and secondary meristem. Actually, what is primary meristem? Primary meristem is directly developed from the pro-meristem. Pro-meristem. Actually, where from it is developed? Pro-meristem. What is pro-meristem? Pro-meristem is the meristem cells present in the embryo. Okay. Embryo of the seed. So that is the first type of cells in the case of the development of plant body. So first type of cells which are present in the embryo known as promeristem. This promeristem will be divided to produce many cells, and some cells will be, and after that produce, suppose you can imagine like that, this is a promeristem cell. This promeristem cell, through mitosis, it will be divided to form two cells. Then again that two cells will be again divided to form 4 cells. Like that, a cell number is increasing, and after formation of some cells, what will happen? Some of the cells will be differentiated. It will be differentiation. What is differentiation? Differentiation means attaining the full property of a cell. That means maturing process. At that time, some special biomolecules are synthesized, such as protein, lipids, carbohydrate, etc. are synthesized in the cell through the various anabolic reactions, and those all molecules are giving the property of its differentiated stage. That means fully developed stage. That full developmental process is known as differentiation. Through differentiation, they will become converted to permanent tissue. Already we studied cells are tissue divided into two categories: meristem tissue and permanent tissue. Permanent tissue. Actually, permanent tissue is a product of meristem tissue. So how that product is formed here? You can see this promeristem divided to form primary meristem. These are primary meristems. Okay, this is promeristem. You can imagine the first cell as promeristem. Then the second two cells you can imagine as primary meristem. Then this primary meristem will be divided to form some more cells, and so most of the cells will be differentiated to form permanent tissue. Okay, these are permanent tissue, and the remaining you can see here one cell. This one cell will be retaining its ability of division. So those cells are differentiated or developed to form the mature condition. That cells cannot divide further. You can see here totally 4 cells. Out of these 4 cells, 3 cells are fully developed or fully differentiated. So they are mature cells, and afterwards such cells will not divide. So such a condition we will call it as permanent tissue. And one cell here, that cell is not differentiating. That cell is not getting full maturity. That means it will be retaining its ability as the primary meristem cell. So this cell we can see this is a reserved cell. Again, that is possessing the ability of division property. So we can call it is a retaining cell of primary meristem. So some cells are retaining its ability of further division. So such cells are known as actually the primary meristem. Okay, so such cells are retaining the property of primary meristem, and that will be there. Again, whenever required, whenever new cells are required, that retained cell will start division. Okay, like that it will be 2, then afterwards it will be 4 in number totally through division process, and again some more cells will be differentiated to form the permanent tissue, and again 1 or 2 cells, few cells will be retaining further property of division, fast division. So again the whole product of cells which are formed through the primary meristem, most of the cells are differentiated, but a few cells are retaining its property of meristem tissue again. So whenever for future purpose, future use, so whenever it is required, again new cells are required, then only that will be starting its further division. So that is very important with regard to the development of primary meristem. Then you can see here promeristem converted to primary meristem, and primary meristem is divided to form many cells. That cells are actually permanent tissue, and some cells are not differentiating, so they are in the state of primary meristem only, and later only they will be dividing according to the use of the plant body. Then next one, what is then you understood actually primary meristem. Primary meristem is directly formed from the embryo. Embryo meristem is promeristem. So promeristem is converted to primary meristem. Then next one is secondary meristem. What is secondary meristem? Secondary meristems are later meristems that will not develop at the initial time of the development, but that will be developed later on at the time of for the matured stage only. So that is why it is known as secondary meristem. So second time only it is developing, not in the initial time. So we will study again. We can classify this primary meristem into 2 categories. So here we have to understand the classification. So first one is primary meristem is divided into apical meristem, and second category of primary meristem is known as intercalary meristem. In order to understand this apical meristem and intercalary, we have to imagine one stem. I can draw this stem here. This is stem, and intermittently here leaves are present. Suppose imagine this is a leaf. This is also leaf. Like that you can imagine. So in this stem, apical means tip. Apical means the tip part. This is stem, so this is shoot system. Okay, and below roots are there. Root also having apex or its tip, that is root tip. This is stem tip. So this is the shoot tip. And here in the case of root tip is there. Okay, anyway, some meristem cells, some part of the meristem, primary meristem cells are present at the tip of shoot and root. Such meristem cells or primary meristem cells are known as apical meristems. So actually this apical meristem and intercalary meristem is based on the position of the meristem. So apical means tip. So apical meristem tissue is present at the apices of shoot and root. So here you can see actually this apical meristem is responsible for the elongation of the stem and elongation of the root. So apical meristem is the primary meristem or basic meristem responsible for the elongation of the plant body, both shoot system and root system. So here whenever these cells are actively divided and producing more cells, more and more cells, at that time what will happen? This stem part will elongate, and some of these cells will be retaining again division capacity. That means it will be calling us meristem further, same type of meristem, and that meristem cell again will divide. So some more cells will be produced, and those cells which are going to differentiate, that cells are putting down, and some cells which are present in the tip, that will be having the meristem property. Again, that will divide. So some cells are formed that will part of the below part. Like that, its length is elongating. So you have to understand, apical meristem is present at the apices of stem, that means shoot, and root. So that is a character, and these are responsible for the main plant body growth. The next one is intercalary meristem. What is intercalary meristem? At the time of division and elongation of the primary plant body, such as a shoot part and a root part, some cells are left behind, and that cells will be that cell may be present at the base of node. Okay, you can see that node means where the leaf is attached. This is, suppose this is now one leaf. This leaf is attached here. So some meristem cells are here present, and this leaf is attached here. This leaf is attached here. So some meristem cells are present here. Like that, here also some cells are present. So we can imagine at the base of, so this is the distance between two nodes. Node is the point where leaf is attached. Okay, here this is node, here this is node. So in between two nodes, some length is there. That length or that part is known as internode. Internode. So at the base of internode, you can see this is internode. This is the base of internode, and this is another internode. You can see, and in this internode, this is the you have to imagine this leaf and this leaf. Okay, so this part is internode, and this is the base of internode. So you can understand, at the base of, or the lower part of the internode will be having some meristem cells. That meristem cell is known as intercalary meristem. Once again, where leaf is started or leaf is protruded from the stem, there some meristem cells are present. That is at the nodal part or at the base of internode. So such meristem cells are known as intercalary meristem. Example, in grasses, if you are, or in all plants, almost all plants, where leaf, if you are taking one stem, shoot part, and you can imagine that leaf attaching point where leaf is attached, and there on the stem some meristem cells are present, that is known as intercalary meristem. Then this apical meristem and intercalary meristem together known as primary meristem. These two types of meristems altogether commonly known as primary meristem. That is a character. So what is a function of this intercalary meristem? So intercalary meristem is present in the internode, base of the internode, and based on the requirement of the plant body, they will be dividing and producing new cells. Sometime and later, whenever the shoot part is getting very old and matured, then it will be disappearing, or it will be merging with the permanent tissue. Then that will not be there. So remember, when intercalary meristem is present at the node or at the base of internode, it is present in the young stages of the development of root and shoot. Young stages of development only intercalary meristem is present. But apical meristem is present in the all stages at the apices of the both part, our shoot part and root part. So it is completely present at the apices region. But the intercalary meristem is present in the young stem and young root only. When the root and shoot part getting older or very matured stage, then these cells, meristem cells, will be merging with or disappearing there, or merging with the permanent tissue. Then that will not be there. So now we are going to see the secondary meristem. So this apical meristem and intercalary meristem together known as primary meristem. Primary meristem is finished. Primary meristem is directly responsible for the growth and development of the plant body. So secondary meristem will be developing later stage only, when the plant body is getting matured. Okay, so that second time some meristem cells are produced in the plant body from the already present matured permanent tissue. Okay, that is very important. Where from the secondary tissue, this secondary meristem is formed. Secondary meristem is formed in the later time of the development only from some permanent tissue. Already we understood that shoot part and root part all made up by tissue. Mainly permanent tissue. These permanent tissues are produced by meristem tissue. But at the later time of development, some permanent tissue, already differentiated permanent tissue, will be undergoing de-differentiation. De-differentiation. De-differentiation means what? This is a specialized cell. This specialized cell is an example of permanent tissue or permanent cell. What will happen? At the later time of development, sometimes this cell with this all speciality may be disappearing. This cell may be there, but these special biomolecules will not, it will disappear. Almost all biomolecule will be disappeared from the cell. So now we can say this is a de-differentiated cell. De-differentiated cell, because it is now it is in the young stage, but before it was in the fully developed stage. This is fully developed stage or matured stage. This matured stage is reconverted to again in the young stage. That property is known as de-differentiation. So this is a young cell now. This is the young cell which is formed through de-differentiation of a matured or developed cell. That matured cell is an example of permanent cell, permanent tissue. So permanent tissue may convert to again the meristem stage through de-differentiation process. And now that newly formed meristem cells are known as secondary meristem, because in the later time only it is formed, and it is formed from the permanent tissue through de-differentiation process. And after coming to the secondary meristem stage, these cells are capable to do fast divisions. Now through mitosis they will be able to produce large number of cells, and after that cell will again may be specialized, maybe getting matured through the production of some biomolecules, special type of biomolecules in that. That means again some cells which are produced from this meristem cell may be differentiated. That process known as redifferentiation. So now this is the meristem. This meristem cell divided to produce many cells, and this cell again may be matured or differentiated. That process is known as redifferentiation. So how secondary meristems are formed? Secondary meristems are formed through de-differentiation from the matured cells or permanent cells, and after coming to the meristem stage through de-differentiation process, that will be able to do fast divisions. So many product cells are formed. Most of the product cells are now again converted to the differentiation process. That process is known as redifferentiation. So first one is de-differentiation, then redifferentiation. Such a process we can see in the case of the development of secondary meristem. Okay, so secondary meristems are formed later, which are responsible. Function of the secondary meristem is production of new cells, which will be increasing the girth of the plant or thickness of the plant body. So we know that this is actually in most of the cases secondary meristems are produced in dicot plants only. Dicot plants only able to do its increasing girth or thickness. So we can understand the secondary meristems are largely produced in dicot plants only. It is absent in monocot plants. That is why monocot plants are not having the ability of increasing its girth or thickness. So this is mainly present in the dicot plants only, but some amount of these secondary meristems are formed in monocot also, because in the monocot we know that if suppose when injury or the plant body is getting wound, at that time what will happen? The already existing cells of that surrounding region will be de-differentiated to form the meristem stage, and again that meristem cells are re-differentiated to compensate that injured part. So that is an example of meristem, secondary meristem development. So at the time of wound development, some meristem cells are formed. That is an example of secondary meristem that will happen or that will develop in the case of monocot plants also. But mainly secondary meristems are developed in the dicot plants only, and that secondary meristems are responsible for increasing the girth or thickness of the plant body. Now we are going to see the further classification of this meristem.